food from a dhaba. So you can very easily understand what is the difference between these two. So I do not know you have tasted the 5-star hotel food for last 9 days. The quality of the training, teaching, everything you have already means habituated almost for these 9 days. Now it is a very tough stuff for a dhaba manager to provide such kind of food to you. So let us see that how much I can do. Okay. So today I am going to discuss about bioinformatics, whatever you have done already. Some of the basic things, some of the concepts. I am seeing some of the students of my own division, some of them have already undergone this class in the normal course, and for them it is a recap, and there are many other new students. So I would like to start with the most important thing in the entire universe, and for that you are here only. The thing is called a Big Bang. That actually gave rise to build up everything in this universe, in this earth. So it was about 14 billion years ago when this Big Bang took place, and then suddenly there are many changes, many chemicals, they started depositing, developing, there are many reactions and other things, and they give rise to a small molecule which we call them as RNA molecule. This RNA are the catalytic RNA, they can replicate by themselves without involving anything. And those catalytic precursor RNA molecule is the ancestor of all the living being in the earth. So from this catalytic molecule, slowly they acquire other different biomolecules, and a kind of primordial, primitive prokaryotic cells developed. And from those things, then with the process of evolution, different life forms in this earth were created. Now, among this earth form, there are many other things, but we, the human being, are the most important civilized and intelligent creature, what we think, in this entire Mother Nature. And we are here to actually see this journey, which was once started at the valley of the river, and now we are talking about the valley of silicon. So we, the best creature of Mother Nature, we are now taking challenge with the Mother Nature, that which took billion of years to develop life form. We are trying to solve the mystery of this biological phenomena just by sitting on a computer with the help of some silicon chips. So let us see the journey. So the journey started from the quest of the life. The people started asking question that what is life, how it was evolved, how all the living beings in this earth are related to each other, what are the elements which carries the information and pass to the next generation. So these were the quest of the life, and to address this quest, the scientific research started. At the earlier phases, when people started work on this to understand these questions of life, like the renowned worker like Charles Darwin, Gregor Johann Mendel, during this time, this was the era of the classical genetics. People started to think about how this genetic information is carrying from one generation to another generation. Then, with the involvement of the molecular different tools like the DNA, it was known that DNA was the genetic material, the structure of the DNA was known, it was known that the DNA, there is a triplet codon that is actually converted into amino acids, and that knowledge is actually developing an era which is called era of molecular genetics. So at this era, what is actually happening? We are trying to understand the cellular biology. We are trying to review the process of the cellular process. And we came to know that DNA, upon transcription with the help of polymerase, transcribed into RNA, RNA with the help of ribosome, translated into form of protein, and this is known as the central dogma of life. So in this era, what is actually happening? The classical genetics, molecular genetics, and cell biology, they merge together to give rise another era. And this era is called era of cellular molecular biology. Now, after this 60s end or the starting of the 70s, we have a very innovative technology development. So the era of technology is started to, we are able to know about the sequence of the DNA, different techniques to fish out the gene like polymerase chain reaction. We are able to clone a gene through the recombinant DNA technology, and so many techniques are now in the fingertips. And due to this technology advancement, what happens? Again, classical genetics, molecular genetics, cell biology, and this technological advancement, they merge together to give us the era of biotechnology. Now, during this phase, the development was not only on the biology field or other field, there was a tremendous development in the field of engineering sciences. So a lot of machines were discovered, developed, and through those machines, now the sequencing of the DNA is very much affordable. So starting from the Sanger and Maxim Gilbert sequencing, we came to different platforms where we can do sequencing, the SMRT sequencing, nanopore sequencing, so many new advanced technology, next generation sequencing technology came. So we can now do sequencing of an entire genome within a couple of days. The first genome sequencing project of human were taken up, and it took at least 10 years time to come up with the first draft genome sequence. But now a human genome we can do within a couple of days. So that much technological advancement was taken due to the engineering development. So that development, what has happened, it reduces the cost of sequencing drastically. It reduces the cost of computing, but it increases the amount of data which was generated through this sequencing. So if you see the graph of the databases, so which were very meager during the end of the 90s, it was suddenly increases in a very logarithmic way due to this discovery of many sequencing technologies. And when we are having a lot of data, because earlier our thinking was that when we came to know that DNA is the genetic material, we are again intelligent creature. We thought that if we know the sequence of a genome, we can know everything. We are able to take challenge with the mother nature, that if we are able to know the sequence, we know everything. But when such kind of huge amount of data was generated, then it was just like this. What is this? Can anyone understand? What is this? Anyone? Nobody knows? It is a hieroglyph which was found in Egypt. Egyptian civilization people use such kind of letters to tell something. So it was a mixture of some drawings actually. But you cannot read this until and unless you have the knowledge of that language, what is written here. So until and unless you know the script, you cannot read that thing. So after generation of so much sequence information and sequence data, it was just looking like a hieroglyphs. It is just matter of A, T, G, C, 4 letters, and combination of these 4 letters, which comprises of sequence. So until and unless we are able to know the language, the way how to read this sequence, how to decode the sequence, it will be impossible to really come out to any knowledge. Only we have gathered information, we have generated information, but we cannot convert it into a knowledge until and unless we know the language. So Once the intelligent creature of the mother nature started thinking that if we do sequencing, we are able to know everything. Now, the same intelligent creator started to think about how different organisms are different. It was our question, and we have generated information sequence, but we are not able to give answer. And if the sequences are there, then how to find out the functions of those proteins? Who will do that thing? How we can relate the sequence and the phenotype? How these sequences are related? Because we need to know how to analyze the sequence, then only we can give the answer. So once it was thought that sequence generation is the major task, so sequence generation is over. Now the next phase is how to analyze the sequence. So people initially thought that I can only do in my lab. It was not possible by few people. They thought that okay, let me collaborate with other scientists or other people of the same department. No, not possible. They thought that okay, the entire university should think on that. No, it was not possible. They tried to think that okay, different universities of a country should think. No, that was also not possible. So again the history repeats. The four major branch of science, it is not university individual, the four major branch of science, they again march together to give rise the era of bioinformatics. So as there are different subjects interlinked, so it is very difficult to understand. So that is why today I am trying to give in a very lucid way what I have understood, okay, about this bioinformatics, and what that knowledge I try to give you, that what is the meaning of the bioinformatics. There are different jargons, terminologies, because there are different subjects interlinked. So that is why my topic of the today's lecture is bioinformatics, the basic concepts and terminologies to understand the molecular phylogeny. So before doing into the details of those terminologies, first let me very much clear that you should understand what bioinformatics is not. The bioinformatics is not just using a computer to speed up the biology. It is not just applying a computer algorithms to biology, and it is not just accountant of the genomic data. Let it be very clear in your mind that if you have some software, you have some sequence, you think that you are doing bioinformatics, it is not. Okay? Then what is bioinformatics? Bioinformatics is the creative use of computer to define and solve biological puzzle. Until and unless with those softwares and tools you are able to define and solve a biological puzzle, that is not bioinformatics. And computer, remember, is only a helper tool to predict, which needs to be tested on bench. So it is only help you to narrow down your search. It is not a decider. You have to tell computer what you want. Otherwise, you give anything in the computer, in the software will give you some result. But it will be very difficult for you to now interpret that result. So before giving anything to the computer, you should be very much clear in your mind what is your question to solve. And according to that, you have to choose what are the different options available in the software. And then only the software will give you a meaningful result which you can interpret. Otherwise, anything if you give, it will develop a phylogenetic tree, it will do anything. It can tell a rice plant is a very close relative to human being. It can tell like that. Okay? So that is up to you, that what kind of data set do you have and what kind of software you need to use for analyzing those data sets. So bioinformatics actually involves three major domains. The first domain is development and implementation of tools and databases, which is major domain of the computer scientist who develop the databases, the tools for different softwares. Second is the development of new algorithm and statistical formulas, which is the major domain of mathematicians, statisticians. They develop those algorithms, and with those algorithms the software people will develop the softwares and databases. And third domain is the use of those tools to generate biological meaningful information and manage them. As a biologist, our major role in this third domain, that we want to use those software for our beneficial purpose. So, we do not need to learn the language, the coding and other things. Rather, if we are able to understand what is our problem and with the available thing, how we can manage those questions, that is the most important thing. So, just like the central dogma of life, in bioinformatics also we have a central dogma of bioinformatics, and that is actually the pillar of the bioinformatics. This central dogma of bioinformatics tells that if two sequences shows similarity, then there are structural similarity. For example, now by this time you are able to know about this kind of terminologies, a query sequence and a match sequence. Query sequence is your query, what sequence you have generated from your own lab. So, that query sequence is matching with a sequence in the database, and this if they are matching, then if we know the match sequence is having a zinc finger like structure, you can expect that the query sequence is also predicted to be a zinc finger like structure. And if there is a structural similarity, then there must be a functional similarity. Okay? So, if you know that the structure of this is a zinc finger, and the zinc finger motifs are majorly present in transcription factors which can bind to the DNA. So, from the sequence directly you can predict that your query sequence predicted to be a transcription factor. Okay. So, without doing the actual bench work, through the sequence you can understand the sequence should have what kind of function. Okay. That is the utility of the bioinformatics, that but definitely you need to test it whether really it is doing that function, that need to be tested before coming to direct conclusion. So, bioinformatics can only predict, can help you to narrow down your search from so much functions, it can narrow down the search. So, let us start with the sequence analysis. We have already, that we may have different types of sequence data. We may have a small sequence data of one gene, we need to analyze one gene sequence data. We may have relatively big sequence data like bigger genes, small genome of viruses. We may have big sequence data like relatively bigger genomes of bacteria and whole complex genome sequence like eukaryotic genome. So, these are different levels of sequence information data which you may have, and for everything you need different sets of softwares to analyze them. So, what do we actually mean by this small and big and other things? So, for example, you have a one gene which you can just with a primer you can generate a read of sequence, and that is a small gene sequence. So, directly from PCR product also you can do a sequencing and generate the result like this, and we may have a big sequence a little bit bigger. So, with one primer it may not cover the entire thing. So, we may need two primers from two ends or couple of more primers, and in that way we generate couple of more reads, and we need to now remove this overlapping portion between these reads and come up with a entire sequence. For that, what we can do, we can just reverse complement this one sequence with the same sequence direction as this, and then we can just overlap understanding, we can do the analysis of the overlap, and we can remove that overlap portion and join them to get the complete sequence. So, this is I am showing only with one, like that there may be many such primers which can join and can generate many reads, and you can generate a sequence. We can generate sequence from the clones. We can clone a gene, a bigger gene, and with the help of the primers which is present in the vector, we can do the sequencing and we can generate the sequence. But all these things are relatively short gene sequence or smaller sequences. We can remove the vector sequence and we can get the gene sequence completely. Now, sequencing of the large DNA, the relatively large DNA, for that we can fragment this large DNA into small pieces, and then with the help of some adapter we can sequence them, and just like the other things we can understand these overlapping sequences, assembly the overlapping DNA sequence, and this is the assembled sequence where this overlap sequence is removed. So, this kind of things is called assembly of sequences, assembly of the reads. Now, whole genome or more bigger genome sequences. So, we now you must have come across with these terminologies, that in those cases we generally used to add some adapters at the two ends of the generated fragments of the genome, and with the help of those adapters we can generate the sequence, and then just like the assembly of the sequence, the reads are assembled, aligned and assembled to form the contigs. So, different reads when we join to generate a larger fragment of sequence, it is called contigs. So, different contigs, in the same way we can join different contigs, we can align, and then we can get a bigger sequence, that is called scaffold. And different scaffold, then again we can do the assembly and we can come out with the entire genome sequence. Okay? So, there are different terminologies. Reads is the first short stretch of sequence. When they are joined together, they are called contigs. Contigs are joined, assembled together to form the scaffold. Scaffold are joined together to form the entire genome. And these are the different contig sequencing assembly program that you cannot do manually. So many fragments will be generated. So, there are several such kind of softwares which are available to do the contig assembly, to join those stretches of sequence to generate the big contig sequence or even contigs to the scaffold. Now, this is the way the sequence company will give you the sequence. Either it is in this fashion, it is called chromatogram, and that will give you that the machine will give you the data of the sequence in this fashion. Also, you can write it like a TGC, and this is the kind of whole genome sequencing, which is the representative sequence of a genome. So, this is the different formats and the sequence file, so which is the data is generated. But after the generation of the sequence, the main aspect is to find out the meaning of the information which is hidden in the sequence. The first thing we need to compare this sequence with other sequences. Until and unless you compare the sequence which you have generated in your lab with other sequences, there is no meaning. How to then find out the function or everything? So, first thing is you need to compare. Now, when you need to compare other sequence, the question is from where I will get those sequence. Okay? So, we will get those sequence, other sequence from sequence databases. Okay? So, these sequence databases are biological databases. They are organized body of persistent data. They have three major tools implemented into those databases. Okay? The first tool integrated into those databases are called update. So, these databases are not a closed database. It is an open-ended database. Every second, every minute, from different parts of the world, people are sequencing different organisms and depositing into the database. Okay? So, it is an open-ended database where individual user can update the sequence. So, there is a portal of update through which you can update the database. Second thing, there is a query component in the database. Through this query, you can ask the database to give you the sequence which you need. Suppose you are working with a fungus or virus or any other organism, you just have to type those name of those genes and the organism. So, the database has built up those kind of algorithms to search in the entire database and tell you that these are the sequences you are asking for. So, they may give thousands of such kind of same sequences. Okay? So, there is a query component, and third one, there is a retrieval component. retrieval is the, you can take out the sequence from the database. Okay. So, all those three things will be part of any sequence database. So, majorly these biological sequence databases are of two type. One is sequence database, one is structural database. In the sequence database, we have nucleotide sequence database and protein sequence database. In structural database, we deposit the structure of different proteins, the 3D structures which were generated through NMR or X-ray crystallography. That 3D coordinates we have to deposit in some format. So, the structure of the proteins are deposited in the database, which is called protein database PDB. PDB means protein database. And the nucleotide sequence database, we have three major general universal sequence databases. One is called National Center for Biotechnology Information, European Molecular Biology Laboratory, and DNA Data Bank of Japan. So, these three are the three major universal sequence database, where any sequence if you generate in your laboratory, it is the duty of you to submit the sequence in the database. Without submission of a sequence, the sequence has no meaning. Okay, you have to deposit the sequence. And there are some protein sequence database like SwissProt and TR-EMBL, translated EMBL and SwissProt. So, they deposit, you can deposit amino acid sequence of a protein into those databases. Now, besides this general sequence databases, we may have specialized sequence database like a Oryza sativa genome sequence database, a human genome sequence database, a Magnaporthy oryzae sequence database. Okay, so, they are called specialized sequence database, but remember that all the sequence information which is present in a specialized sequence database, that has to first present in the general sequence database. So, all the sequence has to be there in this three sequence database. Now, you may have this question that suppose in India, I am working with a particular organism, I have characterized a gene, and I would like to submit the sequence. Where I will submit? In India, no databases is for India. One is from America, NCBI, another one is from a European country, and another from Japan. Then as an Indian researcher, where I should submit? If I will submit to NCBI, my friend who is sitting in Europe, can he access my sequence through EMBL? So, this kind of questions if you have in mind, then I can tell you that in this all three sequence databases, they have a mutual understanding among them that wherever, whenever and whoever submit any data, sequence data in any of the database, within 24 hours, those sequences will be available and accessible from other databases also. But there will be written that this sequence is originally deposited in the NCBI and you are accessing it from the EMBL. Okay. And you can access all the sequence from any of the database. Okay. So, some of the screenshot I would like to show you that this is the NCBI database, then this is the EMBL database, this is the DDBJ database, and SwissProt and TR-EMBL protein database, which is a structural database. So, in those database, you need to first submit the sequence, whatever sequence you generated, you have to submit the sequence. The sequence submission for short stretch of sequence in those databases, they have different portals. For GenBank, which is we call the NCBI sequence submission sequence database, we commonly called as GenBank. Many students do a mistake, they call that them as GenBank. It is not GenBank, it is GenBank. So, GenBank, there are two options, one is called Bankit, another one is called Sequin. So, if you have a short stretch of sequence of few genes, you deposit through Bankit. If you have a larger sequence, for example, a cDNA sequence, cDNA library sequence you need to deposit, you need to use Sequin. Okay. For EMBL, there is only one portal which is called WebAlign. So, in those websites, if you go, you search for those terminology that Bankit or Sequin as per your sequence information, you go to Bankit, you have to register there, you have to create an account, and then there will be several instruction you have to give, and in that way you can submit a sequence and you can update the sequence database. So, this is the Bankit page where you have to sign in, and for that first you have to register only your name, email ID and something, and then you can submit those things. There will be several options, column will be there that you have to give one by one answer, and then you have to submit the sequence. This is the sequence option of the GenBank. Now, when you are submitting a sequence, definitely what I was telling, that you have to give answer of different questions. You have to give something. Only the ATGC sequence if you give, nobody will able to understand again what is this. Okay? So, you need to define your sequence, and for the defining of your sequence, you need to give some criteria, some property of the sequence. Okay? And those things which refers to the commentary or explanation of the information which is appended to a DNA or protein sequence, that is called annotation of the sequence. So, when you are defining a sequence, when you are explaining your sequence, giving some property criteria, then this is called annotation of the sequence. So, you may have heard that many people are telling that first you have to do gene annotation or annotate a gene. So, annotation means you have to give a meaningful information to a gene, what is the property of that gene and other things. So, that common annotation include that source from where you are taking out the sequence, country, organism, protein sequence, predicted protein structure, domains of the protein, quaternary structure of the protein, protein functions, common post-translational modification, any such kind of things which you think that it is relevant to describe your sequence. There are many options which are available in the Bankit or sequence when you are submitting. They will ask one by one this question. So, you have to give, yes, source they are asking, you have to tell from which source you have isolated. They will ask which country, or like that they will, there are options and you have to fulfill those options, and then you submit your sequence. So, you have sequenced a gene or organism and you have deposited the sequence in the database. Next, the question is that why I will submit a sequence, what credit I will get, people will use my sequence, and what is the credit I will get. The credit is that whenever you submit a sequence, the database authority will assign an accession number of that sequence, and that is the proof that that accession number is attached to a sequence which you have generated. So, that is a credit, okay, that I got one accession number for a sequence. Okay. So, that accession number only tells you that you are the owner of the sequence, or you are a discoverer of that sequence. Okay. So, after getting the annotation also, then the question, then what next? The next thing is, now you want to analyze your sequence from the database. Okay. For that, you need to do data retrieval. Okay. So, data retrieval means collection of data from the database. Okay. Now, this retrieval word is not very much familiar to the biologist. Okay. As a biologist, as a common person, we only know a very common word, downloading, because we often do downloading of something from net or in email or something. So, downloading is very common. So, many students, many faculty also have seen that they are telling that you download the sequence from the database, but it is a completely means abusing the term of downloading. You cannot download a sequence from the database. You can only retrieve a sequence from the database. Now, you may ask the question, please let me know the difference between downloading and retrieval. Without that thing, how I will know? So, the difference is that suppose I send you a Word document in your mail, and that Word document may have 25 pages. Okay? And I will tell you, I have sent you a Word document and you download the third page of the Word document. Can you do? can you only download the third page of the Word document into your computer? You can only download the entire content of the file. You cannot download a single page. Okay? But what you can do, you can just click that, and side by side when that preview it will show, you can copy that third page information and paste it in a page. That you can do. Instead of downloading the thing, you just click it, side by side it will show you a preview page. From there you can copy the third page of that file and you can paste it in a new page. So this is the difference between downloading and retrieval. So your database is actually a big file. Okay? You cannot download the entire database. You are trying to download the documents, information which is there in the database. So in the third page of the file is a data. Okay? So you are downloading, you are trying to taking up or collecting the data from the database. And when you are collecting data from the database, the term download is not the correct thing. The term is retrieval. So data can be retrieved, file can be downloaded. Okay? So the database has data, the data you can retrieve, but database downloading is you cannot do by in your local computer. Okay? The entire database downloading. So like this, actually I am trying to tell you that you may have habituated with these terminologies, but you may not know the differences, the small differences, which I would like to tell you actually. So data retrieval, you will able to be surprised to know that the database of NCBI earlier used to give a button to retrieve the data. So a button was there that when you are giving a query that I want this sequence to come up in the screen, so they will show you the screen, those sequences. Now you can retrieve those sequences in your local computer. So there was a button called retrieval of the data, but majority of the biologists, they couldn't able to understand retrieval. Many mails came to the NCBI authority, the web page managers, that I got those sequences I am seeing, but I am not able to download those sequence. Okay? So every time the NCBI asks those people that there is a button called retrieve, you click that and you will able to retrieve the sequence. But again and again, if the people are asking the same question, that actually fed up the GenBank authority also. Now what they have done, instead of retrieval, they have given the download button. Okay? They know that it is very much not correct thing, but what we can do, because for the general public, we use only download, whether it is a data, whether it is a file, both are downloaded. Okay? For Bengalis, we used to tell everything khana, pani bhi khana, khana bhi khana, like that. So data be downloaded, file be downloaded. Okay? So like that, but as a student now in this training program, you should able to tell the correct word. You should not do abusing word like data downloading. You should tell data retrieval and file downloading. Now, why we need to retrieve sequence? So after retrieving the sequence, what is the meaning to, if I retrieve some sequence also, then what? So those sequences after retrieving, we can use to do two things. One is we can understand genomics and we can understand proteomics. What is genomics? Genomics is the understanding of the interactions among the genes, and proteomics means understanding interactions among proteins. Okay? Now genomics is divided into different areas. The one area is called structural genomics, which is the sequence itself. What is the property of the sequence? What is the genome organization? What are the genes which are present in that? What are the warps which are present in that gene? We can designing primer for any detection or any purpose. So that is coming under the structural genomics, where we are only talking about the sequence information and its property. That is the structural genomics. The second component of the genomics is comparative genomics. When we are comparing one sequence with another sequence and come out with some kind of information, like we want to know the annotation of the warp, the relationship and variability between the organism, pathogens and other things, and their strains and molecular phylogeny. So, this kind of things is coming under the comparative genomics. And third thing is functional genomics. When we want to study the function of a gene, function of prediction of the proteins, motifs, blocks, domains, and prediction of the function of the proteins, 3D visualization of the structure. So, all those things comes under the functional genomics. So, all these things I am talking about regarding the bioinformatics, this kind of things you can do to understand the functional genomics or comparative genomics or structural genomics. And in the proteins, we can understand through the bioinformatics the interaction of the proteins with other molecules, and drug designing we can do. We can find out some ligand molecule which can go and bind to a protein and inactivate that protein, and in that way you can discover drugs. Okay? So, this comes under this area. So, retrieval of the sequence is important because we can do so many things after retrieving of the sequence. Now, next thing is that all these things which I talked about, they are actually called data mining. So, what do you mean by data mining? Data mining is generation of the information from the data in the database. So, data is not used in use if you are not generating any information from the data. Okay? So, when you are able to generate information from the data, then it is useful, and the process of generation of the information from the data is called data mining. And data mining are of different types, as I have told you. So, like molecular variability phylogeny, gene finding, primer designing, protein structure and function prediction, promoter identification. So many aspects are there for data mining, and only I am in this class mainly tried to cover the molecular variability and phylogeny and evolution study, that how we can, using the bioinformatics, how we can do this kind of data mining. So, before going to the details, now just give a small break. Now it is interactive session. So, only one way teaching is not the good thing. So, can anyone tell us what is this? Bullet. And what is this? A pistol. So, you know bullet and pistol. I do not know how you know. I do not know. I have never seen. So, but this bullet and this pistol, and what is this? That indicates you know you can see bullet and pistol, but not as a physical thing. Only you have seen the picture of the bullet and pistol. So, this is the nozzle of the pistol through which a bullet is thrown. Okay? So, in this nozzle we have different such kind of grooves. So, when a bullet is passed through a pistol nozzle, it is rotating actually, and that is why when a pistol hits any organism, any body, that creates a small wound, and when it goes out it creates a big hole. Okay? This is the property of the pistol bullet, not the rifle bullet, because they have a grooves and it is moving. Okay? So, it is surprising why I am asking this bullet, pistol, when I am talking about molecular phylogeny. What is the relationship? So, suppose this, if you see this bullet means nozzle, and then you will see they have through the microscope they have such kind of scar mark developed in the both the nozzle inside as well as the bullet. They develop this kind of scar mark because it is revolving inside the nozzle, and it is friction with the nozzle, and it creates some such kind of scar mark in the bullet, and it is coming out. So, this scar mark in very short way, suppose I draw this kind of scar mark which is created after throwing a bullet here, and this scar mark also present in the bullet. Okay? Second bullet also thrown from the same pistol. Now, there are more scar mark added, along with that some more scar mark added. Third bullet will thrown, some more scar mark will added, some may be vanished. And in that way, what happened when so many bullets are thrown from that pistol and the scar marks are generated, what a criminal investigator do? They study these scar marks. And analyzing the scar mark, they can understand whether all these bullets are thrown from a single pistol. Okay? So, in a crime scene there may be 2 bullets, and only a criminal investigator can tell, yes, these 2 bullets actually originate from a single pistol or not. Okay? Again the question is, all this story why I am telling, where is the relevance between the molecular phylogeny and this kind of bullet pistol knowledge? The knowledge is very essential. Suppose the nozzle is a process of evolution, and through this process of evolution what is coming out? They are the variation in the individuals. So, all these bullets are individuals, they have the variations because they are processing out from a nozzle, and by they are originating from the same origin, and they are linked actually, and that linkage is actually the phylogeny, because they have a origin and they are coming out from the same origin. And that is why I have given this example to understand how the process of evolution creates variation in the individual, and even they are variable, as they are originating from the single pistol, the criminal investigator can identify that. As a biologist, as a molecular biologist, you can also do similar criminal investigation by seeing the sequence, that where are those variation and how you can link this variation with their evolution, and how we can tell that whether all this sequence which you are handling, they are coming out from a single origin of the sequence or not. And if they are coming from the single origin, they are related, phylogenetically they are related. So, what is phylogenetics? Phylogenetics is the science of estimation and analyzing the evolutionary relationship. Okay? So, if you want to understand the evolutionary relationship, you need to study phylogenetics. How it is done? It is done through analysis of the DNA sequence or the molecular phylogeny, which we call them, and this helps us to understand the phylogeny of an organism. What is the purpose? The phylogenetic relationship helps us to understand the evolutionary history of an organism and how they are related to others. So, if you want to study that thing, how we have evolved and how we are related, we need to study molecular phylogeny. So, what do we want to know from molecular phylogeny? Whether our sequence has any homology with other sequence, how much similarity is there between our sequence which we have generated in our lab with other sequences which are deposited in the database, and how our sequence is phylogenetically related to other sequence. These 3 basic things we actually wanted to know. Okay? So, again 3 new terminologies are coming up: homology, similarity, and phylogenetically relatedness. So, coming to the first terminology, homology. Those students who have taken the class, I will tell them to be quiet. The other people can tell me what is homology. Any student who has not taken my class earlier, anyone, can you tell what is homology? Nobody knows what is homology? Yes, tell me loud. You are feeling sleepy? Yes, tell. Similar in origin, not in function. Yes. So, that is terminology you have to use, that they should be ancestrally related. Okay? So, homology is the state of a gene or morphological characters that share a common ancestry with a different gene or morphological characters. Okay? So, that is called homology. If the 2 characters, 2 sequence, 2 genes, 2 morphological characters, everything, they are ancestrally related, they are called homologous sequences. So, for molecular sequence data, it is taken to mean that 2 sequences or even 2 characters of within a sequence are descendant from a common ancestor or not. If they are, then the two sequences are called homologous sequence. Now, many times students, faculties, even senior people are also doing a very bad mistake, which I repeatedly I am telling to all the students, you should not, in your future days, you should not do these things. You may have heard many people tells that two sequences were 70% homologous. Many people tells that after sequence analysis they are telling two sequences are 70% homologous. That is not correct thing. Okay? Why not correct thing? Because to tell these things in a very lighter way, I would like to quote the Walter Fitch, who defines homology is just like pregnancy. You are either pregnant or not. Similarly, you can only say the two sequences are either homologous or not. Can anyone say that a woman is 70% pregnant? You can only tell that a woman is pregnant or not pregnant, because pregnancy is a qualitative trait. It is yes or no. Similarly, homology is a state of character. It is not a quantitative character. So you cannot quantify it. You can only tell two sequences are homologous or non-homologous. Okay? Never do this mistake. Never tells that 70% homologous, 80% homologous. Homology cannot be measured. Only we can say whether homology is there or not, but we can measure similarity. Homologous sequence must have similarity, but if there is similarity, we cannot say there is homology. Because two sequences may be similar, but if they are not ancestrally related, they are not homologous sequence. Okay? A virus has a DNA polymerase, a human also has DNA polymerase. Do you think that virus and human originate from same ancestor? No. So, like that, similarity of the sequence does not indicate they are homologous, but if the two sequences are homologous, definitely they should have high level of similarity. Okay? So, these are some basic concept I am trying to give you. You have heard lot of advanced things, but it is very dal roti kind of things of a dhaba, but it will be sustainable food. So, what is homologous gene superfamily? Homologous gene superfamily has two things, orthologous gene and paralogous gene. Okay? So, what is orthologous gene? Example like DNA polymerase of goat, DNA polymerase of human, where the sequences are same, have same function, but found in different taxa. They are originated due to result of lineage transfer during the process of evolution. The genes are transferred among different phylogenetic taxa. Paralogous genes, example like hemoglobin and myoglobin, they found in same taxa but do different, same sequence but do different function. They are called paralogous genes. And these paralogous genes occur due to gene duplication within an organism. Okay? So, we have orthologous gene, we have paralogous gene, and we need to study the homology. Either we need to study the homologous sequence which are of the orthologous gene, or we can study the homologous sequence of the paralogous gene. Okay? Within your organism, target organism which we have done sequencing. So, after the sequence, our major aim is to find out homology. Okay? But we know that we cannot tell homology directly percentage. Okay? So, what we can do? We can find out the similarity or identity, and that will indirectly tell you whether there is ancestral relationship and homology. Okay? Again, two new terms, similarity and identity. To a normal person, it will both are same, similar and identical, same thing or not. If you Google it, that what is the synonym of identical, they will tell similar. Okay? So, but in the bioinformatics term, similarity and identity are different. Okay? How it is different? I am taking now the example of two sequence. This is ATCACA and ATTGAA, two sequence we want to compare. So, if you compare the first two sequence, first one and second one, in this it is AT, in this also it is AT, the last one AA. If this kind of means matching is there, it is called identical matching. Okay. Now, you see this two, where a pyrimidine is there, another pyrimidine is there, both are pyrimidine, here a purine, a purine. So, this kind of sequence is called similar match, okay? And here a pyrimidine and here it is a purine, okay? This is called mismatch, okay? So, as per the bioinformatics terminology, identical sequence means exactly whatever base is there, that has to be present in the other sequence, okay? That is called identical sequence. Now, to do this kind of things, what we are actually doing to analyze the two sequence, whether it is identical or similar or mismatch, how we are actually comparing these two sequence. We are comparing the first base with the first base, second base with the second base, like that. So, what we are actually doing? We are doing positional similarity or identity. We are understanding the position of the base between the two sequence, and that positional identity or similarity we want to study. The positional identity or similarity study is called alignment. So, many times you are hearing this term where we have to do aligning the sequence, alignment of the sequence. What is alignment? Alignment is positional identity or similarity search. Okay? So, alignment is an computational hypothesis which identify positional similarity or identity between the bases or amino acids. Okay? Now, this alignment we can do in two ways. We can do a global alignment, we can do a local alignment. Again, two new terms, global alignment and local alignment. What is local alignment? Local alignment means you have a target sequence and it is the query sequence which we have generated. Just search them to find out where is the maximum stretches where it is matching with a deposited sequence. It is not taking care of from the first nucleotide to the last nucleotide. It is only searching at a glance that in the entire region, this is the stretch of sequence which is matching with a deposited sequence. So, if you are searching like that locally a stretch of sequence with the target sequence, we call them as local alignment search. And if you want to study from the first nucleotide to the last nucleotide of one sequence with the first to the last of the another sequence, that kind of analysis is called global alignment. So, one is local alignment, another one is global alignment. Now, how to align these sequences? For example, we have these two sequences GATTC and GAAATTC. Just if you do a positional study of these two sequences, you can see that there are two match, these two are match, there are three mismatch, and there are two gaps. That means this sequence is two nucleotides shorter than this sequence. So, there are two gaps. Now, for the alignment you need to remember that we are doing alignment to increase the match. If we are able to increase the match, we are calling the alignment is perfect. So, to increase the alignment match, how we can do? These two gaps which is there, we can shift one gap here and we can place one gap here, then you can increase the match. Now, from 2, now there are 3 match. Okay? We can also do like this, we can place these 2 gaps here and you can increase the match is 3, mismatch 0, gap is 2. So, maintaining the 2 gaps, we can align this sequence and increase the match number. Okay? Identical match number we increase. Okay? So, this is the things, but for that in the nature, in the sequence, there is any gap? There is no gap. For our purpose, for doing better alignment, we are creating a gap so that we can get good match. So, in the examination, a student give the examination from his own, may get less mark, and a student may get more marks by cheating. So, where we are doing some cheating to get good match, but here the software will take care of that, that if you do such kind of cheating, you are introducing gap which is not actually there, you are putting a gap, for that you have to give a penalty, okay, and that is called gap penalty. Okay. So, if we open a gap within a sequence, you are opening a gap, that is called open gap penalty, or due to open gap you have to give a open gap penalty. If you are extending that gap, it is called extend gap, and for that you have to give extend gap penalty. So, if you are thinking that through this matching your match is 5, you will get 5 marks, but 1 marks will be deducted from these two, you will actually get 3 marks. Okay. So, for after the alignment, the software will generate a score, and this is the score. How much is the score? You give whatever software will give some gap here and there, and to give a good match, to generate a good value, match value, but along with that it will deduct some marks also. So, ultimately it will generate a score. For one alignment it will generate a score. Okay. So, that score, based on that score we can tell how much is this alignment is perfect. But how to do if the sequence is very large? If it is only this 3-4 nucleotides, we can do some permutation and combination and we can generate a alignment. But if your gene sequence is 1000 nucleotide and if you have hundreds of such sequence, how to do manually? That is not possible. Here is the role of the software. Okay. So, what software will do? It will do through the programming or dynamic algorithm or programming, and it will come through this programming will generate a scores. So, more score indicates more similarity or identity, and it also generates E value. The E value is the probability of not matching your query sequence with other sequence. So, always you will see that statistician cannot show you the nose in the direct way. They will tell where is your nose, they will show the nose is here. So, similarly, they will not directly tell that your sequence is how much is the probability of matching, they will not tell the probability of matching. They will tell probability of not matching a sequence with a target sequence. So, E value indicates probability of not matching. So, if suppose E value of a sequence is 3, E value is the logarithmic value, that means 3 means 10 to the power minus 3. So, 10 to the power minus 3 means 0.0001. So, probability of not matching is 0.0001, that means probability of matching is 99.999. Okay. So, you have to calculate like that. So, lower E value indicates higher matching, higher E value indicates lower matching, more score indicate more matching, less score indicate less matching. So, with the score it is proportional, with the E value it is inversely related. Now, for sequence alignment we can use many tools. Some tools are like BLAST, FASTA, Bleach, Beauty, ClustalW, Mega. So, these are different tools, softwares or web servers through which we can do alignment, and which one important tool is BLAST, which is the first step for any sequence analysis. The BLAST is not bomb blast. The BLAST, the full form is Basic Local Alignment Search Tool. So, as I have told you, local alignment and global alignment. To study the local alignment, BLAST is the tool which do the alignment in the entire database with your sequence immediately, with based on the high probability of matching area, and it will generate the score value and E value. So, BLAST is the algorithm used by family of 5 programs that align your query sequence against the sequence in the database, and statistical methods are applied to judge the significance of these matches, alignments are reported on the order of their significance, and it is estimated by the applied statistics. So, BLAST has different options like BLAST N, which compares a nucleotide query sequence with a nucleotide sequence database. So, if you want to search a DNA sequence, you want to ask the database that my DNA sequence, whatever I have generated, is matching with any known DNA sequence in the database, then you have to click the BLAST N option. Okay, so it will tell you that whether your DNA sequence is matching with any known DNA sequence in the database. Next thing is BLAST P. BLAST P, if you have generated a protein sequence, amino acid sequence. So, then you have to click on the BLAST P option, and you have to give the query as the amino acid sequence. Then the amino acid sequence of your sequence and the database amino acid sequence, they will match, and they will tell you whether your protein sequence is matching with any other protein sequence deposited in the database. Now, you have a DNA sequence, you want to know whether my DNA sequence is matching with any protein sequence, because the DNA ultimately codes for any protein, suppose, okay, and whether that DNA sequence codes for any protein sequence, you want to know the protein product of that gene. For that, you have to do BLAST X. So, what it will do? It will first convert the nucleotide sequence into 6 different frames of coding sequence, protein sequence, and then they will analyze that with the deposited protein sequence. Okay? So, that is called BLAST X, and last one is T BLAST N, where you have a protein sequence, amino acid sequence. You want to know which gene is actually coding this protein. You want to know about the gene, you have a protein sequence. So, you have to do the T BLAST N, where it will tell you that your protein sequence is matching with this kind of gene sequence. So, that indicates that this is the gene which actually codes for this protein. So, the data you have to give in this BLAST, you have to give the data in this format, and the result will come out like that. As I am telling you that there is a score value and E value. So, it will generate based on the significance. The highest score means this, with this particular entity your sequence is showing highest similarity, and E value less means it is highly probable that this is the probability of matching of the sequence, low E value. But here you can see it is also giving maximum identity, sequence identity also, but we should not take that as a sequence identity, because BLAST is doing a local alignment, it is not doing a global alignment. As it is doing a local alignment, so the problem is that local alignment cannot tell you the entire feature of the sequence. So, from BLAST what we can know, we are able to know the sequences that share similarity with the query sequence, and it helps us to retrieve those sequences from the database. But through the BLAST we cannot know the quantify, or we cannot quantify the sequence similarity, because it is a local alignment, and it cannot tell us the relationship between the sequence. So, it can only helps us to retrieve the sequence which is showing high similarity. So, what actually I try to tell you, suppose here I am showing you the BLAST result. It is showing one query sequence with one subject sequence, it is showing the alignment, and it is showing that out of 373 nucleotide, 346 nucleotide are showing similarity or identity. So, that indicates 92% is showing identity. Okay? Now, this is the situation, this is your target sequence, and in your query this region are matching, and that is why it is almost covering the entire region, and it is telling you 92%. Now, if you scroll down, you will see such kind of sequence where 84 nucleotide out of that 346, actually the sequence, only 84 stretch of that sequence, out of that also only 77 is matching. That means the situation, this is the target sequence which is deposited, and whatever sequence you have given, only it is matching with this portion, a stretch of sequence, not the entire. And in this also you can see it is 91%. Okay? Because it is a local alignment, So, just based on the percent similarity, because it is calculating the similarity in the region where it is showing the similarity. So, you cannot tell that it is really that much percent similarity is there between the true sequence or not. To understand the real sequence identity, we need to do global alignment, and for this global alignment we need to do using some other softwares like Mega or many other softwares are there. So, which do a quick pairwise alignment, calculate the distance metric like that. They will do one by one alignment, and then you just see sequence 1 and 2, sequence 2 and 3, like 1 and 3, 1 and 4. So, individual pairwise alignment, they will generate score, and from this score they try to understand that which score is very close together. So, based on that they will create a guide tree, and from this guide tree they do a progressive alignment, one by one sequence they will take up, and then they will following this guide tree, then they will do a multiple alignment, global alignment. So, this is a global alignment, start from first nucleotide to last. So, this is a global alignment. From this global alignment we can then generate a sequence identity matrix. So, only after the global alignment we can generate such kind of sequence identity matrix, which will tell you how much percent sequence identity is there. And also from this global alignment we can generate this kind of phylogenetic tree to establish the relationship. So, until and unless we do a global alignment, we cannot tell the percent sequence identity and the phylogenetic relationship. So, what is a phylogenetic tree? Phylogenetic tree is looking like a kind of branching of lines. So, it has a root, it has some nodes, and these nodes representing different taxonomic units. It has different branches which represents the relationship, and within a taxonomic group, if there are different branches, they are the sister taxa, and this is the distance in a branch which is called the length, which is actually the genetic distance. So, this comprise of a phylogenetic tree. Sometimes in the phylogenetic tree, at the point of this node you get some kind of numbers. This number is called bootstrap value, which defines the significance of this tree, or the tree is how much stout enough that these two branches should hold together. The branching pattern of a tree is called topology, and the representation of the relationship through line is called dendrogram. Okay? So, this is a tree topology, and this is the line representation of the relationship is dendrogram. So, type of dendrogram, if you see, because when you use any software, this kind of things will come up, because software will have all this type of options. Until and unless you know about what is the meaning of those things, it will be very difficult for you to draw a phylogenetic tree. And if you depend upon computer, it will be a disaster. Computer can generate any type of tree. Whatever you tell, computer will generate, but you cannot interpret them. Okay? So, it depends upon what is your query, and you have to tell the software that I want this type of tree. So, type of tree, there are two types, one is called phylogram, one is called cladogram. Phylogram and cladogram, both of this tree can be represented by rooted tree or unrooted tree. So, again 4 new terminologies: phylogram, cladogram, rooted tree, unrooted tree. I am making you feeling bored? You are getting something, because for last 9 days you are handling with all this kind of terminologies, I hope, or not. You have developed phylogenetic tree, you have developed the phylogram or cladogram. Phylogram you have developed, not cladogram. Why not cladogram? Why phylogram? Your data is telling you, asking you about the phylogram development, or it is by default you are doing? There is the problem. If your data sheet is not asking you to develop a phylogram, why you will develop phylogram? Have you asked any time? No, because you do not know what is phylogram and what is cladogram. So, first let me introduce about those 4 terminologies. What is phylogram? Phylogram is a phylogenetic tree that indicates the relationship between the taxa and also conveys a sense of time or rate of evolution. The temporal aspect of a phylogram is missing from a cladogram. So, if your data set has a sense of time or sense of evolution to tell, then you should use phylogram. If your data set do not have any time frame, do not use phylogram. Clear? Okay? In your thesis or anywhere, if you want to develop a phylogenetic tree, first you look at your data set, whether your data set is conveying a time that during this phase million years ago, this was the sequence and slowly it is evolving like that. Are you trying to tell this kind of evolutionary story based on a time? If yes, do phylogram. If no, do not do phylogram. Okay? So, that depends upon what is your query. Again I am telling you, you have to tell computer what you want. If you depend upon computer, computer will generate by default anything that you cannot explain. So, this is the phylogram, this type of phylogram. Okay? The feature of phylogram is that you can see here these branches, somewhere it is going long, somewhere it is short, somewhere it is so much branch pattern, somewhere it is less branch pattern. What does it mean? How we can infer a conclusion from this kind of tree? Tree will be generated by software. Until and unless you can interpret the tree, what is the need to do? Okay. So, you have to understand what is the meaning of this kind of different patterns of branching. So, if you see in a phylogram that something is going ahead of other things, that indicates this is a clade or this is a group which is evolutionary more advanced, because you have generated a phylogram. Definitely it has a sense of time. Okay? So, that grouping indicates this is evolutionary more advanced, and as there is a time, you can calculate this much distance means how much time, because there you have to give the data that always you will see that in case of phylogram there is a small bar which is generated at the bottom, which indicates that this much unit indicates this much year. Okay. So, if this is the thing, so a small 1 centimeter bar may be representing 1 million years, or it may be 10 years, that depends upon again your data. So, if such kind of things is coming, you can tell that they are evolutionary more advanced. In this kind of condition, here after a certain, so this branch, this distance is a genetical distance, it is a time. So, after some time it is evolving. Here it is not evolving, continuously it is going, only after so many years it has evolved into 2, not more. So, that indicates this branch, whatever the entities are there in this branch, they are evolutionary more conserved, because they are not having any mutation, they are not evolving faster. Okay? And here where more branches are there, that indicates they are evolutionary more diverse, more changes, continuously it is changing. Okay? So, if your data set, you try to explain your data set in this fashion, then only you use phylogram, otherwise not. Clear? Now, what is cladogram? Cladogram is the same dendrogram, where this temporal concept is missing, it is not having any time sense. As it is not having time sense, what is your expectation, how the tree should look like? It is trimmed in, all the branches should be trimmed at a point. Okay? So, there is nothing is coming out or something like that. So, all these things is coming in a straight line, in a trimmed fashion. Okay? So, that is called rectangular cladogram or phenogram. There is another type of cladogram which is called slanted cladogram. So, again a new terminology, a slanted cladogram. So, in the software when you do, you may have done and you will see this kind of all options are there, slanted cladogram, rectangular cladogram, circular cladogram, so many kind of things. But you need to know what are those things and whether that is required for your purpose. If it is not required, if you give, software will generate anything, but you cannot interpret them. Okay? So, slanted cladogram and rectangular cladogram, where you have to use slanted cladogram, where you have to use rectangular cladogram. So, for example, when we want to do grouping for taxonomic studies, we want only some taxonomic studies, that these are the different groups, these groups are related, these groups are distantly related. If your questions or hypothesis is to explain that, then you use this kind of rectangular cladogram. And if you want to know the convergence, divergence, parallelism in the evolutionary studies, then you use this kind of slanted cladogram. Again some new thing came, that convergence, divergence, parallelism. What does it mean? So, if you see, these are the converging sequence. So, in the process of evolution, it may be possible they have separately evolved and then they have doing so much evolution, mutating, one day in future they may converge. So, these are the convergence. So, these are parallelism, all these things are evolving parallelly. Okay? And this is divergence. So, in the evolutionary process it is going, then diverged out, it is going, then diverged out. So, if your data, through this data you are trying to explain your data in this fashion, then you use slanted cladogram. Again I am telling, do not do anything for the beautification purpose. I have seen many students, many theses came to me, beautiful circular tera sab hila hua hai tree. What for? We do not know. They ultimately is doing that, these are the groups and they are related, they are not related, only they are telling like this. But they are doing a circular cladogram, they are doing this kind of slanted cladogram for what purpose? If you are not going to explain anything on this aspect, do not do. Okay? Because if you are trying to publish such kind of things, the reviewer will ask you this question, that what for you have generated a slanted cladogram. Your data you are not analyzing as those kind of things. Okay? So, do not do unnecessary thing in science. Then two things again, rooted cladogram and unrooted cladogram. Both phylogram and cladogram can be rooted, can be non-rooted. Okay? Now, what is rooted and unrooted? So, this is a rooted cladogram, okay, where we assume that all the taxa which we are analyzing, they are originating from a common ancestor, a common thing. For example, as I am working on viruses, I can tell that, for example, a different tomato leaf curl virus. It is definitely expected that different tomato leaf curl virus should come out from one original tomato leaf curl virus, and there are different isolates of tomato leaf curl virus. So, they definitely should have a root. So, if your data set, you also have such kind of sequence where you are assuming already that all those sequences should originate from a single sequence, single ancestor, then you give a root. Okay? But if you suppose you want to do a fungus sequence, a bacterial sequence, a viral sequence, a human sequence, a plant sequence, anything, so never you cannot think that they all are coming from one ancestor. So, there is no root. Okay? So, in those cases you use a unrooted dendrogram. Unrooted dendrogram do not have any kind of root, and that is why in the software you will see some kind of circular trees are also there. So, those circular trees are unrooted tree. So, if your data set, suppose you are working on Magna porthygrisae, 10 isolates of Magna porthygrisae. So, and you are doing a circular tree, what is the meaning? All the 10 isolates should originate from one isolate of Magna porthygrisae only, and you are telling that no, I am assuming they are not originated from single thing. So, this completely your assumption and your result is totally different. Okay? So, do not do those kind of unnecessary things in your research. Okay. So, it is assuming that all taxa derived from a common ancestor, it assumes that all the taxa do not have a common ancestor. Now, these are types of tree I talked about. Now, coming to how to develop a tree, or what are the methods to develop a tree. So, there are two methods, one is called character based method and another one is called distance based method. Okay. So, what is character based method? Suppose you have these two sequence, you want to do a tree or analyze these two sequence through a phylogenetic tree. How to choose a method? Now, in these two sequence, what you are seeing that there is only one nucleotide difference. The other nucleotides are identical. Okay? Only there is one nucleotide difference. So, one character in the sequence is different. Okay? Under that condition, you generate a matrix and you can give the score as 1, because one nucleotide is varying, other things are same. That is why you can give as score as 1, and from there you generate matrix. So, this is one pair of sequence, in that way several pairs of sequence you have to analyze, you have to generate scores, you have to generate matrix, and then you have to generate the tree. So, as it is based on a character, then it is called character based tree. Now, the character based tree again of two types, where in one condition all the sequences are highly similar. You are taking different strains of a virus, they are highly similar, only few nucleotides here and there they will be varying. Okay? You are taking another set of sequence where you are trying to find out that there is only one or two sequences are matching among these sequences. There are two diverse sequences. That even there is no similarity identity, only few nucleotide here and there they are matching, rest of the things are not matching. That is maybe a condition, a highly diverse sequence or a highly conserved sequence, highly similar sequence. So, in both these cases, either you have to find out the sequence which is dissimilar, develop a dissimilarity index matrix, or you find out a similar nucleotide which is only similar, to create a similarity matrix, okay, and develop the tree. So, in both this condition you use character based method. Okay. Now, what is distance based method? For example, I have taken these three sequences. Here the same thing, that it is differentiating by one nucleotide. So, score is 1. In the third sequence again it is T, in just like the first sequence it is T, T is T. So, you can tell that these two sequences are totally identical, but it may happen that during the process of evolution it was earlier. So, based on that this two score is 0, because there is no difference, but it may happen that earlier it was G, in the second generation there is a mutation, the G is converted to T. Okay. So, one generation has passed. Okay. So, what happens in this condition, the score may be 0, but there is a genetic distance, there is a genetical distance which is 1. Okay. So, if such kind of situation is there where we need to consider about the genetical distance also, in those conditions you have to use distance based method, create the distance matrix, develop the tree. So, it may sound a little weird now after few slides it will be much more clear. So, there are two types of character based approach which is available in many softwares. One approach is called maximum parsimony or minimum evolution, second approach is called maximum likelihood. So, in the softwares if you have analyzed, you might have seen this kind of options, maximum parsimony, minimum evolution tree or maximum likelihood algorithms, they are written, that phylogenetic tree. So, again you have to choose that which type of algorithm I have to choose for my data set analysis. Nobody will tell you, computer cannot tell you. Okay. You have to decide which type of algorithm you have to use, whether it is maximum parsimony, whether it is maximum likelihood. Now, where we have to use maximum parsimony? When sequences are quite similar, small numbers of sequence. In those kind of condition, we have to use maximum parsimony. If the sequences are more diverse, computationally more intense, and we need to analyze the mutations, such kind of condition you use maximum likelihood. Okay. So, if you have in your data set 10 isolates of the same organism, which type of tree algorithm you should choose? 10 isolates of same organism, which type of algorithm? Maximum parsimony. If you have different isolates of different organisms, no, that is now you have to tell that when to then apply maximum likelihood or other things, that I am coming then. Okay. So, now try to understand the character based method more clearly with an example. I would like to show you the example of a gene sequence of human, rabbit and chicken. These 3 individual organisms we have taken: human, rabbit and chicken. The gene is a orthologous gene, and there are only few nucleotides variation in that gene. So, after developing, so this is 2 human rabbit, human chicken, rabbit chicken, these are the pairwise alignment, and this is the score which has been generated from this alignment, that there between human rabbit 1 change. So, their score is 1, between human and chicken there are 3 changes, so score is 3, between rabbit and chicken 3 changes, so rabbit and chicken it is 3. So, it is called matrix. So, in that way matrix has to be generated, first alignment has to be done, then matrix has to be generated, and then based on the matrix which one is coming closer, human and rabbit. Okay. So, it will take human and rabbit closer 0.5 and 0.5, and then it has to add with the chicken, then 1.5, then 1 and 0.5. So, this 3 will be between human and chicken, and 0.5, 1 and 1.5, this relationship with chicken and rabbit. So, this is called a tree. Okay. So, software will do based on that. First they will do the alignment, then they will generate a score and the matrix, and then from this matrix they will generate a tree. So, that thing is happening inside the computer, inside the software. You do not have to think about that. Only you have to tell the software whether you need a character based method or you need a distance based method, you need a maximum likelihood or you need maximum parsimony, that kind of things you need to give the command. The rest of this kind of things the software will do. Now, I would like to tell one example of distance based method. One example of the algorithm is called neighborhood joining or neighbor joining approach or algorithm. Here suppose we have created the software after the pairwise alignment, it created, this is a matrix. So, we are analyzing the spinach, rice, mosquito, monkey, human, this kind of different sequence, one gene sequence, and so it is a matrix, whatever is there it will be there also. So, we are trying to see spinach and spinach 0, rice and rice 0, like that, and we want to see spinach and rice, this is the distance matrix. Okay. So, distance means they are distant. So, from this distance matrix first the software will find out which one has the less distance. So, they have find out that between the human and monkey they have the least distance. The distance sequence, the matrix distance is less between human and monkey. So, what software will do? They will first, in the first step they will join the human and monkey, and it will form a human monkey cluster clade. Okay. Then human monkey as a group they will do a progressive alignment. They will do a progressive alignment and they will try to find out what is the relationship between this human monkey as a group with rice, spinach and mosquito. Okay. And that will come to the next cycle and it will generate a next distance matrix. Here you can see between the monkey human as a group, the the mosquito is showing the least distant. Okay? That means after the monkey human as a group, it is coming closer with the mosquito. So you can now join the mosquito with this monkey human group and create another clade which is mosquito monkey human is a clade. So this is one clade completely. There are two branches deviating and then another branches which is coming out. So in that way you are creating the tree. So now again with this mosquito human monkey you are trying to establish the relationship between spinach and rice. And when you do the next level of cycle, now you observe that it is not showing that with mosquito monkey and human as a group there is least similarity, rather the least similarity is between rice and spinach. Okay? So what you have to do? Then you have to create another clade. Okay? So this is a clade, this is, so there is not a monophyletic tree, it is a polyphyletic clade. There are two different clades has been created, one is this, another one is this, and finally you have to join these two to create this final tree where there will be two cluster, one is this spinach rice cluster, another is mosquito monkey human big cluster. Within this cluster there will be another monkey and human, this cluster. So there is close relatedness, phylogenetically relatedness between human and monkey, followed by the mosquito, and they are distantly related with this another clade, where within these two they are again closely related. Okay? So in that way you have to explain that how using different algorithms you can create a tree. This creation is done by the software within in the background, they will tell you only the tree. So after generating the tree you need to do bootstrapping. Bootstrapping is a method for assessing the statistical significance of the position of the branches in a phylogenetic tree. So it is a kind of horoscope matching. When people are doing horoscope matching for marriage purpose, what for? To test the significance that a boy and girl can stay together in the branch or not. Okay? So that is the test of significance, that computer is telling that these two sequences should stay together in within a clade. Okay? Now you have to do a horoscope matching, that if horoscope matching will generate yes, they are having very good match. So if there is a second arrival of a girl or a boy, they will not separate it out. Okay? So a new sequence if you include, they should not separate out. So to test that significance, you have to do some statistical analysis, and that analysis is the bootstrapping. That is a statistical analysis for each aligned pair. It samples scores from random position of the alignment, adding the score. When all the pairs have been sampled, it converts the scores to the distances and computes a tree. This whole process is repeated many times, and the frequency with which the particular tree features are observed is taken as a measure of the probability that the feature is correct. So what actually we do? So these are the three sequence, these are the different position. So first they will create a guide tree, and guide tree is telling that these two should have some probability of staying together. Now how it will do? It will analyze three replication. So you can see the first nucleotide, then it will take fifth nucleotide, then it will take sixth nucleotide, like that. It is not 1, 2, 9. So in randomly it changes those nucleotides and then again formulate the tree and try to see that how many times, suppose 100 times they have done like this kind of replication, how many times the sequence A and B should come together. So out of 100 times there is 70 times they are coming together. Okay? So that tells you the significance or means confidence of the tree, that whether the two sequences should stay together. Okay? If it is significance is 20 only, then any sequence you take up another sequence, that those two branch will break down and they will join with another group. Okay? So to test the significance of your tree, you have to do bootstrapping. Then only you can tell that this is a phylogenetic group or this is a clade which should stay together. The entity within this taxa, within this group should stay together. So, by this time I think you have many confusions, okay, but I tell you, you should not have any confusion. So, what you have to do actually, you choose a set of related sequence based on your data, you choose a data set, okay. Then you obtain the multiple alignment. After obtaining the multiple alignment with any software, you ask a question, is there strong sequence similarity or identity, which it is telling after the multiple alignment you definitely have to generate a sequence identity matrix. So, from that sequence identity matrix, can you tell that within all the sequences they have a strong sequence identity, okay, like more than 90%, 95%? If that is there, if it is, the answer is yes, through the analysis if you see yes, there are high similarity or identity, if yes, go for maximum parsimony approach, okay. Then do bootstrapping and generate the phylogenetic tree. If the answer is no, no, we do not have 95% similarity, then the question is there recognizable sequence similarity in that sequence identity matrix, that it may be ranges from 60 to 80% or 60 to 70%, or it is 30 to 20 to 30% in that range. So, whether it is a recognizable sequence similarity or identity, if the answer is yes, go for distance matrix, okay. Do bootstrapping and develop phylogenetic tree. If the question answer is no, that no, I do not have any recognizable similarity, only 10%, 20% is so less sequence similarity I am getting, then you go for maximum likelihood, again a character based approach, do the bootstrapping and develop the phylogenetic tree, okay. So, depending upon your choice, you have to choose which method of phylogenetic algorithm you have to use. Based on your data, you have to choose which type of phylogenetic tree you have to create, whether it is a phylogram, whether it is a cladogram, a slanted cladogram or a rectangular cladogram, or whether you need a rooted tree or unrooted tree, everything depends upon your data set. And always remember one thing, majority times I have observed that the students started analyzing the sequence, you have a sequence of 500 nucleotide and the deposited sequence, because you have not done the full genome sequence, full gene sequence, it may be a partial portion of a gene, but in the database the sequence is deposited for the full gene, and you have taken up the full gene, your sequence is 500, the deposited sequence is 2000, and you are trying to analyze those two sequences, nothing coming out, okay. Software will give you some result, no problem. Always remember that whenever you do start doing sequence, the first thing is that length of the sequence, it should be similar. I am not telling that if it is 502 nucleotide, always everything is 502. It may be go around 510 nucleotides idhar udhar, but it is not that one is 500, another is 2000. Okay? So, always choose the same length of the sequence, then only all these things will happen, otherwise nothing will happen. Okay? So, the software will tell that your sequence is a totally different sequence, it is a new novel sequence, nowhere it is found. Okay? So, from where we have started, that in those way, using the silicon chip, we are trying to solve this biological mystery which Mother Nature was created, and it takes millions of years to diversify those organisms, to establish the phylogenetic relationship, and we, the best creature of Mother Nature, we are trying to solve the molecular phylogeny by just sitting in a computer with some softwares. So, we do not know who will win. So, we have many limitations, like bioinformatics can only predict, many times it gives erratic results, different subjects are interlinked, so understanding is difficult. It may be possible to make models, hypotheses, but in reality it may not appropriate. So, who will tell the last answer? Our beloved Mother Nature or a silicon chip? With this point to ponder over, I thank each and every one of you for your kind attention. Thank you. Any question, query, clarification?
open to all. I think the lecture is all about the molecular phylogenetic tree, the basics. The last but one slide was the amazing one. Hope you have taken the picture. What kind of algorithm you have to use under what circumstances? Very, very important point. And he also made several points, but the couple of points I want to re-emphasize. The length of the sequence must be same. But having said that, it is not only the length must be same, but also the position of the base in that particular coordinate also must be same. Is it clear with all of you? Did you understand this statement?
Not understood?
The gene sequences are there in the genome. When somebody is sequencing the genome, every base is allotted a number. That number is popularly called as coordinates. That is also called as genome coordinates. Right? And if you are sequencing a gene, for example, active gene you are sequencing, right, from your lines. So you get the sequence, maybe 500, 600, 800 base pair. When you are mapping or blasting the sequence to the database, it matches with something else in that database. When you do that, you can notice these coordinates. Especially when you are blasting the whole genome, the coordinates will come right in front of you. Right? Note that coordinate. Right? And that coordinate is very, very important, because 800 base pair is fine. But if you are taking another entry, having 800 base pair, starting from another coordinate, the tree will be different because the nucleotide sequence will vary. You understand? So, the point what he emphasized was, there should not be any human error here that would reflect in the form of some nodes in the tree. You can do it yourself. I think now you are quite aware of the things now. You take 600 base pair sequences from 10 isolates of an organism. You remove a couple of bases in deliberately, 1 or 2 entries, you make a tree. And you keep all the bases and make a tree. You will understand how the tree topology changes the moment you remove 1 or 2 bases. That would have a far-reaching implications in the interpretation. This is a common mistake most of the students they do. We have noted also, we have observed also. But things are not going the way, at least I expected, maybe everyone expected. But henceforth, my suggestion is, whenever you do a molecular phylogenetic tree, keep these points in your mind. Okay? Go back to the last week's story, entry, data curation, removal of poor base calls, keeping only the high quality base calls. Right, all those things are very, very important. What you are doing in the process, you are removing the so-called sequencing errors. Right? Sometimes the machine may read instead of C, it may read at A. So it's a sequencing error. You cannot call it as a kind of mutation. If you do that, then it may, sometimes the interpretation goes in a different direction, you know. You will make a far-reaching conclusions. I found a new gene with a single point mutation in a particular position. You make it all claims. So these are all some of the very, very basic things. Very often people don't do the way it is really needed. Right? Those 2 points are very important, and I'm sure the lecture is very informative and loaded with a lot of information in terms of learning, for learning. So thank you, Anirban. It's an excellent lecture. I was sitting in the back and listening to this lecture, and try to follow whatever directions he presented in this lecture. Okay. Thank you very much. And if you have any quick queries, we can have those queries here, as well as we are having the tea also. Let's do that. Okay. Yeah. Any queries? Maybe two questions we'll ask, huh? Because the tea is waiting outside. Yeah. Anybody? Okay.
No question?
Yeah.
Okay.
No question. Still, you can interact. With him. He is going to be here for during the tea time as well. Okay. So if there are any questions, sir? Okay.
I just want to say, he's not going to give you any marks. Okay. You can always ask him questions.
Yeah, it's not questions, rather we wanted to test how much you really understood. So this morning a lot of questions, interesting questions came and I'm sure he put things very simple and elegant manner. In the same word, in a lucid manner he presented. Certainly most of the things are presented very straightforward, right? So thank you Anirban for a wonderful lecture, and I'm sure the students will remember this lecture forever and keep it up. Excellent. So a token of affection to you. I call upon Dr. Ramakrishnan to present this a small memento. It's not a very big memento, but it's what we could offer. So please come this side. Some of you can take pictures also.
They want me to stand in front
Okay.
of you.
Okay. Yes.
Thank you very much. Thank you.
Thank you.
Excellent.
Deliver a nice lecture. Thank you.
Excellent. Keep it up. Yeah, great. Now let's go for
सर स्ट्रीम बंद करें अभी
fundamental over study. No, prayer resolution is okay. It's not about the method of resolution. It is about the type of sample. What is recognized as a microbiome, which can be preserved as a compound.
So whatever sample we are taking, we can like preserve the sample as such, and after a certain time, in like transplantation, we can go on reviving that again and.
Okay, I'll put it straight away. We all know that a gram of rhizosphere soil, there are 10 to 7 to 10 to 8 culturable bacterial count. Okay, if you go to rhizosphere, we have 10 to 5 to 10 to 6 per gram of leaf. If you go to endosphere, you have around to 10 to 4, sometimes 10 to 5 culturable bacteria itself. Right, it is living there. Correct. And this we call as a term it as a microbiome, right? It may encompass not only bacteria but also other associated microbial communities like the eukaryotic one, protist, microfauna. The question is how to preserve it. As of now, I'm not encountering any literatures on microbiome preservation anywhere. Yes, please. Give a mic to him.
I also didn't see any literature, but in your lecture once you said while phyllosphere washing with buffer, you said you washed with the phyllosphere microbes and then you centrifuged it and diluted it in 1 ml and whatever you use and
Exactly.
the next you use it.
Yeah, I'm glad that you listened to that. When you extract the microbiome from the sample, there are two kinds of extraction. One is you extract everything using a repeated washing and pooling all the wash, whatever you are getting. I also told per gram, you can go up to 50 ml of buffer. You do 5 to 6 rounds of washing, serial washing. You end up getting around 300 ml of the total wash. You go for a kind of centrifugation, pellet everything, and resuspend in a small volume, and you divide into different small liquids, and one of the liquids ensure that they are the true representative of each other in the sense of the total volume, and you can go for a preservation using some methods, maybe glycerol preservation or maybe some kind of DMSO assisted preservation. I see this one of the techniques. But it is not going to retain the viability of every microbial cell which is present in the microbiome. Right? Certain cells may not survive. For example, wombats, they won't survive. Suppose the microbiome consists of some zoospores of the lower fungi, they may not survive. Protists may not survive. But the DNA will be. Alright? So, microbiome, if you consider the genes, the gene contents, this method is likely to work. This is one of the methods, keep in your mind if you want to really preserve microbiomes as a long-term strategy. This washing and followed by centrifugation and reducing the volume and adding some preservative, and you can preserve it for a long time in my research. Okay, then what about the preservability of this particular microbiome? There is always a challenge. I already told you, certain organisms, bacteria for example, it survives long time in glycerol. If you are keeping the temperature, maintaining the temperature of -80 without any break, you can recover it whenever you want. But what about other associated microbial communities? You may not get it. There are some deficiencies. To circumvent this particular issue, what I am thinking, we can have parallel approach. For example, you plated out the what you found after 48 hours. crowded colonies of bacteria you found. Correct. It's a bacterial lawn, right? And this bacterial lawn appeared on the plate because those bacteria could utilize whatever nutrient supply in the medium. But again, it is not representing the entire microbial diversity, less than 1%. We all know that. Okay. But still, I would consider that strategy as preservation of a culturable microbiome. Understand? So, what you do, you go for a minimal media that would bring, that may not select or that may not be biased towards certain microbial communities. It gives an equal opportunity for most of the organisms to grow. It might bring a lot more diversity on the plate. If you are using a nutrient-rich media, that nutrient richness might favor a few microbial communities. In the process, the actual diversity may be lost. So the best way forward is you use different kinds of media by changing the nutritional composition. Try to bring as much of diversity as possible. In the first dilution itself, whatever is appearing, you collect the lawn, and as it is, you can go for the preservation without losing anything. So, in the later point of time, because we have the technologies to know what is there, you can go for a profiling, right? So use that as a stock of everything mixed together, correct? Whatever appear on the plate. So the idea is the relative abundance of different organisms might vary. The one which is fast growing and rapidly colonizing things may appear. The slow growers may have been smothered by the fast growers. But still, I would say that there may be a kind of opportunity for everybody to grow. And this is one way we can preserve it. So when you talk about this, this is something completely deviant from the conventional microbiological and pathological techniques what we have been comfortable with. We always go for isolation. The word isolation means what? You isolate an individual organism. But here what I am using the word isolation is isolation of the microbiomes from the environment. Separating the microbiome from the environment. So that means you separate the culturable organisms from the environment. Then you try to preserve it. I think these two methods can go in parallel, at least in the absence of any well-known technique. It might offer a temporary solution for preserving microbiome. Any other ideas in your own perspective? These two are, it's riding in my mind. One is extract, cereal washing, extract it and preserve it. Okay. Of course, this is mostly the one which can be detachable, detached from the environment. For example, epiphytes. Maybe the one which is colonizing the rhizosphere, maybe in the bulk soil. What about the one which is living deep-seated, for example, in the pipes? There is no other option. Right? For endophyte preservation, the best way forward is you preserve the whole plant. That's the only way. But as a long-term strategy, I'm not sure it is going to be useful for any kind of analysis. That remains to be seen. Okay? But again, you think it in your own angle, in how many different methods we can preserve microbiome as a stock, as a genetic stock. Why I am suggesting this point, I think this morning there was a talk about endangered microflora, microflora is not the right word, endangered microorganisms. So what do you mean by endangered microorganisms? The microorganisms which is on the verge of extinction. And the fact remains, there are so many organisms are even before human interception. They appear and they extinct also. That is always happening. But let us not bother about all those things. We will bother about the one which is there in the environment for us to explore. Those organisms we can preserve in this method as a long-term strategy. Right? We all know that the microbial content and microbial composition in an environment is largely decided by the so-called environmental factors, right? If you irrigate the soil, we all know that there would be a spurt of microbial activity, and the microbial contents, quantity, composition, abundance, everything changes. The same soil, it is going a drying phase after a few months, you will see something else here. You may wonder where they are going. Motile organisms may migrate towards a better place where they can survive. Those who cannot move on their own, probably they may form a kind of a street structures and they may lead its life as a sedentary life, or they may die also. Right? All possible scenarios are there. But the point is, at that particular experimental time, if you collect that sample and preserve it as a long-term strategy, I think you can go back in time after 10 years and you can analyze what was there as a microbial content a decade back or 2 decades back. This is something nobody has done it in the past, right? Since the new technologies and new opportunities are emerging, and the new ideas and the new methods of biodiversity preservation has to come in. So microbiome preservation, I'm sure it's the next big thing that is going to happen, right? This is one of the discussion points. I think we can really discuss in the larger gatherings how to preserve the microbiome for future people to use it. Okay. Yeah, this is. But again, you can think, and if you have some points, you can put it in the group also. We can explore that. Anybody else want to add before we take up the practical what we planned today? And let us not forget, one theory is still left. So no time today. Today, here I have too much of information since morning. So we will take up the lecture tomorrow morning. Any observations? The microbiome preservation is really a point we need to really discuss. Okay, please think. And by the way, we preserve microbiome in our lab, and we have microbiome preserved. It's almost 6 years now. 6 years of 10 years. Some years with us. That came from very inaccessible places in the country. Port Blair, Andamans, Almora, small volumes. Anyway, let's take the class forward. The lecture what I want to deliver is on my biological microbiome and an unexplored microbial diversity tomorrow. Today we will have a very, very important practical. The team is ready. Annotation, gene annotation using some of the online pipelines or online programs. I think I was told that the sequences have been already shared with you. Probably you might have already retrieved from the or recovered from the or downloaded whatever, and start handling that. And I think Shanu will tell some few steps how to assign a protein to a sequence what you got from your sequencing efforts. Remember, whenever you are approaching, please listen carefully, whenever you are approaching a database or submission of gene sequences other than ribosomal or RNA or IPS, which is having a molecular taxonomic value, all other gene sequences, you cannot get an accession number unless and until you supply the protein which is likely to be coded by the sequence. So that means whatever sequences you are generating in the lab need to be annotated, and annotation means you are trying to identify the proteins which is potentially encoded by that sequence, and using plastic key analysis you need to identify whether it is the same gene what you really wanted to sequence, right? And many times you may encounter a problem. One of the major problems is what is called as internal stop codons, and those who have done it, you might have already experienced that. IS ICs, internal stop codons, right? What do you mean by internal stop codons? Anybody heard about this? Raise your hand, those who heard about it. No? You heard about it. Okay. I think very few people have heard about it. It's nothing but suddenly after a few amino acids it says that it is stopped. That is not the one correct. Okay. We have online tools. There is no magic bullet here. Online tools are available and you just run your program here. Remember, a small rule, when you do the sequencing, if you do the proper curation without any compromise, you will not get any internal stop codons. If you don't do the curation properly, then there are chances that you may end up with internal stop codons. That's a trick, right? So that means you have to curate your sequences properly. You have to remove the sequences which is not originating from the sample, rather it is introduced by the machine, and that differentiation you have to make, whether it is from the sample or it is from the machine. If it is from the machine, then it is a problem. So you have to identify using different methods, what we call triangulations, repeat sequencing, go for the forward rule sequence, compare them, and identify the correct base call based on the quality values. If you do all those things, perhaps you will end up getting correct sequence which is there in the native state. And if you use that material as a starting material for annotation purpose, most often you will end up getting a correct group. So you will not have any problems. Okay? So the point is very clear. Any bioinformatic tool, you have to devote time. If you cannot devote time, don't take up this kind of problems. Why? If you cannot spare time, and if you jump steps, and you will be polluting the databases with the wrong sequences. Nowadays, that will not be possible because they are asking you to submit a protein codes, protein sequences. That means unless the sequence is correct, protein will not come. So this is a recent, I think last 10 years they have integrated this. Before that, people were simply uploading the sequences and generating that. It's no longer it is possible. Okay. So without protein code, they are not going to assign the accession number. Okay. So these are all some of the nitty-gritties of submitting sequences in GenBank, especially for the protein coding genes. And yeah. Shielded. And again, the story is same. Whenever you are submitting species in the GenBank also, same story goes. There the annotation is for taxonomical purpose, for example, species annotation. You are assigning the species name to a sequence what you generate, and unless and until the sequence is correctly annotated, the database staff will approach you. They will tell that no, what you submitted is not Pseudomonas fulva, it is Pseudomonas protegens. Please address it. These kind of checkpoints have now come. So, meaning the recent sequences what is being uploaded are quite dependable and quite high quality. Right? This is very, very important. So, I make it a clear point here, unless and until you are sure about the identity of the organisms, don't call your organisms with that name. You need to have a proper confidence over the data sets to call a species a species. And maybe different kind of pressures will come. This is submission, that is so many. Then you hurriedly wrap up the thing and not bother whoever is going to face it, let them face it. You can do that, and people are doing that. So under that circumstances, politely you call it as a species rather than wrongly calling Pseudomonas fluorescens as Pseudomonas putida, Pseudomonas putida as Pseudomonas eliginosa. All these things are happening, right? You have to be very careful because taxonomy is very important. Okay, and there are instances, there are some organisms which was in the commercial domain has been withdrawn now, especially in the case of Pseudomonas. Let us not forget that. Why it is withdrawn? Species identity is not after 15 years or 20 years of marketing that stream, one fine morning one fellow came and told it is not fluorescence, it is universal, and at the highest level in the government system they took a decision not to promote it. So we did. So this is also happening in our country, right? So why this happened? Because no attention was given to the taxonomic identity of an organism. There are too many things are there in the name. Name matters. Okay. Okay. Please, Shanu will. You are going to connect your laptop here. Good. So, all right, and she will tell the annotation, importance of annotation, the relevance of annotation, not only for the gene but also for the taxonomic identity of the organisms what you are handling. Okay. But she will take up the gene annotation. Yeah. This practical, don't hurry it up. There is no cut-off time like 5:30 to leave. There is no compulsion, right? So you can take as much time as possible. Complete the annotation part and whatever, and share the results in the group. Thank you.
Okay, here. ध्यान रखना। Hello everyone. I hope you guys have downloaded the sequence file which I shared in the WhatsApp group. All of you have done that? Okay, that's nice. Okay, that file contains up to 20 sequences. Okay, and each sequence is specific to a particular organism and its gene. Okay, that sequence was, last day I taught you how to design primers for up to 1 kb of product. That is what I did for those sequences also. So they are up to 1 kb of the sequence length. Okay, so the sequences which I have given you are the curated sequences. Okay, what has any one of you ever done the sequence submission? Okay, which software you have used? Which database? NCBI. Through? Okay, 16S sequences. That is direct submission. You don't require any protein sequence for that. Okay. You can do direct submission of conserved regions. In the cases of bacteria, 16S. In the cases of fungi, ITS. Okay, but when you design primer for gene, you have to give protein sequences. Okay, for GenBank, the tool which you use for submission of the sequences is Bankit. Questions, a series where you will submit the sequence, then they will ask you who's the author, so many attributes are there, and at the end there is a table they will ask you for feature table. What that feature table includes is what is the length of your gene, what is the length of your mRNA, what is the length of your CDS, what is the start codon. Okay, why do you need that start codon? Can anyone tell me? Yeah, we need to tell that from that particular point, that start codon which I am going to give is having ATG. That means it will code methionine. From that particular point, the expression, the translation of protein starts for my particular sequence. Okay, so what happens is we have our sequences and we don't know, okay, from which codon my reading is starting for translation of protein. Has anyone known how many reading frames are there in a particular sequence? Okay, do you know what is a codon? Okay, what is codon? Can anyone tell? Just anyone? Three bases which can code a particular amino acid, exactly. So, okay, for starting of protein translation, when it starts, which amino acid is considered? Methionine. And does okay, so in bacteria, methionine I have taken at ATG, I have considered. Yeah, amino acid is different. Yeah. So, but ATG I consider that. Okay, that is your start amino acid. Okay, and what are the codons for stop translation? Does anyone know? Three are there. Mainly three are there. I generally use a term like UA, UGA, UG, like UAA, UGA, UAG. Okay, this is what you can learn it easily. You can remember it also. So what we are going to do today is, you have the sequences and we are going to retrieve that what is the start codon in that particular gene. Okay, you have your sequences. The tool which we are going to use is XPECI. So don't get confused. XPECI is a very big software. It's not just for translation only. So when you are going to use it for your purpose, just write XPECI translate tool in Google. XPECI can be used for varying purposes. Here we are going to use just the translate tool for that. Is it there? Okay. A sequence can be read bidirectional, like 5 to 3 and 3 to 5, right? Okay. When you are reading the sequence, there are 3 frames, in a direction. 3 reading frames can be formed. Okay, reading frames mean, do I need to explain that also? Can I? Do you want me to? Okay. Do you guys have sequences? Okay. First reading frame. Do you know what is reading frame? Okay, no. Okay, I'll explain. The first codon. What is the first codon here? In the first sequence. TGA. Okay. Second codon. AGG. Next codon. TTC. That is one reading frame. Now you skip the first base and start reading it again. GAA. Next GGT. Next TCC. That is your second reading frame. Now just like that, you're going to skip the first 2 bases. Now AAG, GTT, CCC, 3 bases we count in a codon, 3 reading frames. Understood now? Okay. So when the translation happens, XPC is going to give you the same type of result, okay? It will give you 3 reading frames for 5 to 3 prime direction and 3 reading frames for 3 to 5 direction, okay? Now you can copy your sequence, any sequence, it's your choice. I've given you multiple sequences. Okay. Before clicking on translate, I would like you to focus on the output format it has provided us. Okay. The verb boss, it will tell you the stop codons, the spaces between the residues. The second is the composed, that means it will tell you, okay, from which bases are absent and all that stuff. If space is there, it will not, consider that. Okay. The next is, it will include nucleotide sequences also and your amino acids also. And the last one, nucleotide, amino acid and no spaces. We are going to see all the 4 type of outputs, okay? Let's start from verbose. One more option is there, the genetic code. XPC works along with NCBI, okay? So just see how many options it has provided us. So if your gene is very much specific to any one of those, you can select that particular and go for it. Here I am going to go for the standard option. Okay? Just click on translate. Go down. You can see how it is showing. Here frame 1, frame 2, frame 3 for 5 to 3 direction, and the next is 3 to 5 direction, frame 1, 2 and one more is there, 3. Relating? Okay, good. Any confusion till now? I hope not. Good. Now here you can see only bases, you can see only the bases. You cannot see amino acids here. So, what am I going to do? I'm going. Nope. What is not there in this type of output? Stop codon is also not there. Huh? Can you see stop here? No, huh? Stop is not there, huh? Right? Can you see in the verbose? Sorry, in the compact? No, right? Let's go to the third option. Don't forget why we are using XPC. What is the purpose here? To see what is the start codon and to see that if my reading frame is having stop codon or not. That is the purpose, okay? What am I having here now? On the third option. Here we have it. Can we see? Are you guys with me? Okay. Can you relate to what we just discussed? The reading frame, how it came to be K amino acid here. AAG will code for K. So this is the third frame. Okay? Shall I proceed next? What is the next part? Okay, nice, good. Now to see what is the start codon, you have to count the amino acids. Okay? Wherever position methionine is coming, that M is coming, that is your start codon, and you are going to give that particular number while submitting your sequences in the GenBank in the feature table. Is it okay? Okay, good. Sometimes it might happen to you that you are getting some discontinuous reading frames. What to do in that case? We are going to give frames in fragments only. Has anyone seen while retrieving data from database that they give you some ranges when you download a particular gene sequence? Has anyone noticed that before? It generally happens because of that only. Because the reading frames is fragmented, so it will give you ranges from this particular base to this particular base. It happens like that sometimes. Even sometimes it happens like this, that particular sequence, that particular range might code for some other protein and the second range might code for some other protein. It happens like that also. So you have to be very specific while you are submitting your start codon. You should be very sure about your sequence. Now, to check whether this sequence, whether this reading frame is right or not, what we can do? You click on this M here. It will take you to one more page. Can you see here? What are the informations we have here? It will tell you the frame and everything. Okay? You can copy the sequence from here, this nucleotide sequence, and you can just paste it in the NCBI and see that whether the gene for which you have selected that particular reading frame, it is going to code for that particular gene or not, to just verify whether the sequence is going to code for that particular protein or not. Okay? This FASTA format which you have right here, you can copy that and use protein blast, not nucleotide blast. Huh? There are different type of blasts, if you guys know, nucleotide and blast P, blast N. Use blast P for this. So now I have given you almost, I guess, more than 20 sequences. So you guys can play with it. And if you find any query, any question, then you can ask me.
Yeah, what she explained was how to assign a protein sequence for a nucleotide sequence. You have to play a bit with different options. It does a six-frame translation. It does. I'm sure one of the frames must be correct. Anyway, practice it. Share all your results group-wise. Same group. WhatsApp group. We'll see that. Any doubts here? Anybody? And not necessarily this sequence. You can retrieve some other sequence from the database and you can try it. It works. No issues. Any group-wise you send group-wise or together?
No, sir, together.
Which group will be addressing what? Any idea?
If they are facing query with any particular
Okay,
sequence,
you can try all the 20. Why not? Okay. You practice all the 20 each group. You practice all the 20. integrated, then that would open up different kind of channels. You can use an uncurated sequence also and try it out, how it works. So I already told the sequence is properly curated without any error, then you end up getting correct equation. Alright. If that is not, then you get these problems. So everything goes through sequence quality only. If sequence quality is good, and you will get a correct protein. Right. And I recall the practical what the Neo and the Leibniz team handled here. Genome annotation. Do you remember that practical? Same principle. Here you are handling with only one gene. There we are handling a genome.
A genome. Yes, sir.
Otherwise, fundamentally these two are the same logics. If you can continue this practical for another half an hour here, there's no stopping. Complete it and go because tomorrow we may not have to do this. Complete it, generate the protein sequences of all those 20 secreted sequence share with you and share in the group wise you share in the WhatsApp. I have few announcements to make. Tomorrow we have a lecture from 9 o'clock. Okay. That lecture is a sponsored lecture from Eppendorf. They will be here at 8:45 itself. Okay. So again, tomorrow's lecture is not at 10 o'clock. It is at
9.
Say it loudly.
9.
Let's meet at 9 o'clock tomorrow here. Okay. But you can continue this practical so that there is no spillover because already one lecture is carried forward. We are carried forward into tomorrow. Let us not do that. This practical is complete and go. I have already informed Pradeep of it. Okay. Yeah. So best wishes. So you be here. Them complete. Okay.
Okay. Are you close side? नहीं नहीं होगा